then let's get started. So, hello, my name is Stephanie Ishu. I am the Manager of Research Communications and Marketing at Target ALS, and today we'll be going inside the mission of the organization to discuss the challenges of ALS research and how Target ALS is breaking down those barriers to find effective treatments for ALS. So, I'm joined today by Target ALS CEO Manish Raisinghani and our Senior Director of Scientific Programs, Amy Easton, and I'm going to let each of them introduce themselves. So, Manish, can you start us off?
Sure. Well, hello, everyone. Warm welcome to all of you who have taken the time to join us today. I'm Manish Raisinghani. I have the privilege of serving as CEO of Target ALS. Before Target ALS, I was in the biotech industry. I worked as a scientist and then at a stint in marketing. And then I was looking for something purposeful to do and have been lucky to be part of Target ALS since its beginning in 2013.
Thanks, Manish and Amy.
Hi, everyone. Welcome to the webinar today. So happy to be here. I am Amy Easton, the Senior Director of Scientific Programs for Target ALS. I joined in 2022 after 20 years in drug discovery, spent quite a bit of time at Bristol Myers Squibb, and after that, Genentech, which is where I really started to dig into research on ALS and decided that I wanted to really commit all of my time to discovering new therapeutics to treat ALS and have been really on this mission since my early years, probably since the time I was 18, knew that this is what I wanted to do and have been studying neurodegenerative disease all of my career.
Thanks, Amy. We're lucky to have you both with us today and on the leadership of the organization. So, to kick things off, I'd like to start with the unfortunate reason why we're all here, ALS. So, Amy, could you tell us a little bit more about the disease and what it is?
Sure, thanks, Stephanie. So, amyotrophic lateral sclerosis, or ALS, is a progressive degeneration of upper motor neurons in the brain and lower motor neurons in the spinal cord that communicate with muscles throughout the body to control voluntary muscle movement. So, these neurons are important for allowing people to initiate movement, control their muscles, so that we can do all of those daily activities that we do: walk, type on our computers, speak, swallow, and even breathe. Compared to other neurodegenerative diseases, ALS progresses quickly from the time of diagnosis. This can be within 2 to 5 years, and it's always fatal. So, for about 10% of people with ALS, there is a known genetic cause, and this is typically inherited and passed down through family members, affecting many generations of families. For about 90% of the other folks who are diagnosed with ALS, the cause is still really unknown. What we do know is that there have been identified about 40 different possible genetic risk factors for ALS, as well as potential environmental factors, and that a combination of two or more genetic factors or genetic and environmental factors can really lead to the onset of disease. So, what you're hearing is that there's not one cause with one cure, but there are many different causes and potential treatments for different patient subpopulations.
Thanks, Amy. ALS is clearly a really complex disease, and complex problems need robust solutions, which is why we're here with Target ALS. So, Manish, could you tell us a little bit more with your viewpoint as someone who's been with the organization from the beginning? What is Target ALS and what's our mission?
Sure. So when we started in 2013, and since then, our overarching mission has always been to find effective treatments for ALS, so that everyone with ALS can live a long quality life. And we never used the word cure because we felt our goal would be more realistic if it was looking at effective treatments first. And we recognized early on that to find new treatments, we needed to find new drug targets. So what are drug targets? Drug targets could be a gene, a RNA molecule, a protein, a molecular pathway that one modifies so that one can alter the course of the disease to benefit the person facing ALS. And so from the very beginning, we knew if we can find good drug targets, which means these are drug targets which are backed by a strong understanding of biology of the disease, we will maximize chances for the success in future clinical trials. I think all of us have heard that over 90% of clinical trials fail. So it's really important that we take the best shots at the goal because even if we have a drug target that fails, but we know its biology well, we will learn something which will inform future clinical trials. But if we have drug targets which are not based on a strong understanding of biology, we lose that chance as well. So in fact, our name Target ALS really refers to the how we refer to a target in the world of science, as I just described. So that's where our name comes from.
Thanks, Manish. And so we talk a lot about barriers, and unfortunately, progress has been slow for this very deserving community. And so we talk about breaking down barriers at Target ALS, and there are scientific barriers that the disease presents to the research community, as well as systemic barriers that plague medical research overall, and specifically within the ALS research community as well. Amy, could you please lead us into a couple scientific barriers that ALS presents and how Target ALS is breaking those down?
Yeah, thanks, Stephanie, for that question. When you look across the research landscape for ALS, we've identified three main areas of research where there are significant barriers that are slowing down our ability to get treatments to patients. And so the first is really the study of the fundamental aspects of the disease biology. So, like I mentioned earlier, this is a heterogeneous disease, meaning that there are different causes that can lead to different cellular pathologies in the brain and spinal cord across patients. And even when we do know that an individual has a specific mutation, we don't fully understand the cascade of biological events that can lead to motor neuron loss. So we've started to put some of the puzzle pieces together, but we're still continuing to find new pieces so that we can really understand the full picture. And we're also trying to put those pieces into temporal order so that we can understand what is the primary and earliest event that leads to motor neuron loss. So when you study a complex disease like this, the number of experiments that one scientist could do in one lab could become infinite, and it can really slow down the process. So this is what Target ALS is doing to help facilitate that. First, we dedicate about a third of our overall budget to funding multidisciplinary consortia working together to study the underlying cause of the disease. And we're also providing those researchers with critical tools and resources. So specifically, we've been investing in building large repositories of patient samples. So both postmortem tissue and biofluid samples from people living with ALS. They're well characterized and well preserved to enable high quality research by the community. Um, there was a time when these samples were scarce, and so scientists were left to using different tools to study the disease, like imperfect mouse models or imperfect cell lines that didn't represent what happens in neurons or what happens in the human condition. So we're really filling that gap to provide a key resource to the community to study this human disease. Finally, we're leveraging state-of-the-art technology to conduct multi-omic profiling of all of the samples we have. So these new technologies allow us to detect changes in genes and proteins in patient samples that we wouldn't have been able to detect otherwise. Even 5 years ago, some of this technology didn't exist. Things like long read sequencing that I can talk more about later, single cell sequencing, all of these things provide high resolution and broad analyses of all the genes and all the proteins in tissue or in samples. And so we share these data out through Target ALS's data engine freely to researchers worldwide to access the data. And so again, these are really wonderful ways to help spur understanding basic biology and breaking down the barriers that make it hard to study. The second area of research that we're really focused on breaking down barriers to is identifying biomarkers. So a biomarker is a measure of a particular biological function. So for example, if you go into the doctor, Stephanie, you might go for your annual checkup and the doctor measures your blood pressure or your cholesterol levels in your blood. And these are biomarkers of heart function. And so these, if they're changed, they can indicate that there might be a problem and that you need to be treated. So this doesn't exist for ALS patients yet today. We're working very hard to fund research and do all the things that we can do to push forward a diagnostic biomarker because this really provides physicians the confidence to diagnose ALS early and then allow patients to get on treatment as early as possible to prevent motor neurons from dying. And given that ALS is a heterogeneous disease, we also need disease stratification biomarkers. And so what I mean is biomarkers that tell us which sub-patient populations would benefit most from which particular therapy. And finally, it takes time and specialized technology to get biomarker assays that have high specificity for the protein or pathological protein of interest and high sensitivity. So we can detect even small levels of the pathological proteins in blood, for example. So we have many ways to break down barriers to facilitate biomarker discovery. The first is really funding multidisciplinary consortia, often combinations of drug companies and academics working together to identify and validate new biomarkers for ALS. And one of those key steps to validating the biomarkers is that you really need access to human CSF, cerebrospinal fluid, which bathes the brain and contains proteins that can indicate what's happening in the brain, or blood samples. And so we've been actively collecting those biofluids from our ongoing natural history study to enable this resource. And we share these samples freely for our researchers to study. And finally, we also fund special initiatives to generate reagents that can be used to develop highly sensitive biomarker assays. And we also collaborate with other nonprofits and even FNIH to develop a biomarker toolkit that we can put in place ultimately in clinical practice. And the third area that we're working to break down barriers in is really that whole drug discovery process. So it can take a long time from the first idea of a new drug to FDA approval, and it can easily take even 20 years for this to happen. So, for example, Qalsody or tofersen really started with a genetic finding that mutations in the SOD1 gene can cause ALS back in 1993. And then with the advent of new technology, antisense oligonucleotide technology, small pieces of DNA that can be designed to target pathological RNA. With the advent of this in the 2000s, Ionis and Biogen were able to develop the first therapy to treat those SOD1 mutation carriers. This was tested in the clinic first in 2014, and they found that really there were some properties that they needed to improve. So they went back to the drawing board, more potent ASO with better properties, which ultimately became Qalsody, which was approved in 2023 and is the successful drug that it is today, showing meaningful benefit to patients that take that drug. So you can hear that this can take a long time. Target ALS is really removing the barriers to rapidly accelerate the drug discovery process. And we're doing that by first providing human data that allows researchers in industry to have confidence in their drug target and confidence that they know how to modulate it. Do I want to inhibit it? Do I want to activate it? What is the right therapeutic way to manipulate this? And so we're, again, sharing all of that data out through the data engine. Also, they need tools and they need preclinical models that can help them test their drugs to be sure that they're doing what they ought to do. So we provide them access to both animal models and human motor neurons to be able to do this. You have to remember that industry, in many cases, just doesn't have the infrastructure to breed mouse models or to maintain human neurons on a large scale. So we're giving them the opportunity to do this. And finally, it does take time and technical expertise to create a drug that you would actually give to patients, meaning that it crosses the blood-brain barrier, distributes well to the affected areas of the brain and spinal cord, and that it's safe. So we do a number of things, like I mentioned, not just sharing out data through the data engine, but that's a really wonderful way to jumpstart drug discovery programs. But we fund collaborative consortia between industry and academics to do this work so that they can share their expertise and models. And we even recently provided a tailored funding call to support technical innovation for development of the next wave of gene therapies so that we can really facilitate technical improvements to allow them to cross the blood-brain barrier. And so those are the three areas of research that we're really working to break down all the barriers that exist there to facilitate new development of drugs.
Thanks, Amy, for that comprehensive answer. And of course, as we said earlier, there are those scientific issues that we're looking to solve, but those scientific issues exist within a system, right? And that's what Target ALS is hoping to put speed into with our innovation ecosystem. So, Manish, could you tell us a little bit more about those systemic barriers and how Target ALS is looking to solve them?
Happy to. And I think I'll be touching on some of the themes that Amy has referenced already. But again, from when we started in 2013, our first objective was to understand the barriers that were in the way of progress for ALS research. And then the whole approach we took was to try to address and overcome these barriers. Now, we have overcome a lot of them, but then certain barriers sort of endure and want us to continue to work on them. So, as we have really scaled up our ecosystem exponentially over the last couple of years, these barriers have uh, revealed themselves again in different scenarios. So, Amy mentioned that we fund collaborative projects, and what we had recognized early on was that the inertia for the research community in academia certainly is to work in their own labs by themselves and not collaborate so much. And this is even more so when it comes to a collaboration between academia and pharma biotech industry. And that's why from the very beginning we insisted that to get Target ALS funding, scientists had to come together, leverage complementary expertise to get our funding. And this was not just people who were best friends or famous names, they truly had to show complementarity of expertise. Now, as we have scaled up our ecosystem, we are bringing in new talents and technologies from around the world, from new scientific disciplines, from other disease areas who are not used to this culture that we have built in our ecosystem of collaboration. So, again, that's a silo that one has to continue to break down as one moves forward so that a much larger community can participate and learn that a collaborative approach is the only way that one can accelerate towards finding treatments for this disease. The second point I'll make is again, Amy referenced the fact that we provide access to research tools and resources to scientists worldwide. And this is really critical because if we really want to expand the ALS research community, encourage emerging generation to work on the disease, for pharma biotech to work on the disease, if you want to draw in experts from other disease areas to work on ALS, we need to lower barriers so they can pursue their ideas, they can work on ALS. And what this really does is and has done is it democratizes ideation from so anyone anywhere in the world can work on ALS by accessing these tools and resources irrespective of whether they're funded by Target ALS. And the other key piece in this is that all these tools are accessible quickly with no reach through on any intellectual property generated from their use and with nominal cost typically of shipping and handling. So all of this contributes in accelerating the work. The third thing is if we want to support the best ideas, we need to have a system that is credible, that a scientist anywhere in the world knows and is confident that their idea will get a fair and transparent evaluation from Target ALS. So it is not because they are famous themselves or they are friends with someone on Target ALS team or with a board member. So we use what we call an independent review committee mechanism to make all our funding decisions. And this is a group of scientists by design, 50% from academia, 50% from industry, who make all our funding decisions. And it's completely firewalled process from anyone on the team or board. And that allows us to, again, draw in the best talents, technologies, and ideas from around the world to move the science forward.
Thanks, Manish. Target ALS seems to be trying to cut whatever red tape's in the way that's preventing progress from happening as quickly as it can be. And so we were really lucky that all of you guys on the call submitted really great questions, and I see some more coming through in the Q&A. So I'd love to dedicate the rest of this time to really getting at the heart of being a resource for the community here. So one question that came up a few times, what is the current treatment landscape and what are the most promising breakthroughs in research? Amy, would you be able to tell us a little bit more about that?
Sure, I'd be happy to. So, right now, there are probably about 60 different active clinical trials ongoing, and they're testing a number of different mechanisms. But what I want to share is what I'm most excited about on the horizon, which is probably in the next year, you're going to start to see therapeutics that are targeting TDP-43 or somewhere along the TDP-43 pathway to prevent motor neuron degeneration. And so the reason I'm excited about this is that TDP-43 pathology is a hallmark of ALS. So about 97% of people diagnosed with ALS have this pathology that accumulates in their brain and in their spinal cord. And so what we think is that this pathology, like current dogma is that this is occurring early. And so that if you can actually modify TDP-43 and reduce this pathological accumulation, um, that you can actually intervene to prevent motor neurons from dying at all. So that's the excitement about targeting TDP-43. And what Target ALS has been doing is to fund a number of different approaches. So there are many different forms of pathological TDP-43. There's loss of function in the nucleus, aggregation in the cytoplasm, which causes a number of different changes in normal basic cellular function. And so there are different therapeutic approaches to tackle TDP-43, including downstream events like changes in RNA biogenesis. So there's ongoing clinical trials to target these so-called cryptic exons that form when TDP-43 can't do its normal function of correct RNA processing. You create these aberrant pieces of RNA called cryptic exons that just have lost their function, and or they result in proteins that don't normally exist and also can be pathological. So so I'm kind of including all of those different pieces of the TDP-43 pathway when I say I'm excited about this new wave of therapeutics that are coming in. I also am excited about alternative approaches. So TDP-43 is important. We need to see how those clinical trials pan out so we can inform what therapeutics come next. But also alternative approaches are really important. And I think coming from the folks that we're funding, along with other researchers that are in clinical trials, some of the interesting things relate to understanding better the role of the adaptive immune system, the role of T cells and B cells, and how they're impacting disease onset and progression, as well as the role of the innate immune system. So, immune cells that reside in the brain that are meant to protect your brain from infection, and how modulating them might be beneficial.
Thanks, Amy. Manish, do you have anything to add about the current treatment landscape and breakthroughs that are happening in the field?
The only thing I would like to do is just reiterate the point I had made earlier that the reality, unfortunately, in the world of science is, and when it comes to clinical trials, is that close to 90% fail. And so it's really, really important that we make sure, as far as is possible, that these clinical trials are based on drugs that have a strong basis in understanding biology of the disease, because that is the only way we can maximize chance of success. And we should we recognize that we have in the entire space of ALS, there is only a limited amount of resources we have when it comes to financial resources or the most precious resource, which is the human resource, people with ALS who can participate in clinical trials. So we want to be really careful in what types of clinical trials are launched. And because that will, again, we need to make sure we maximize the chance of success for every attempt that we take.
Thanks, Manish. And Amy, so you talked a lot about TDP-43. There have been a couple questions that have come in about it, being that it is this hallmark of disease in ALS. Seems that the researchers are focused on it because it is that, 97% of cases. Are there similarities to amyloid and tau in Alzheimer's disease drug development?
Yeah, thanks, Stephanie. I think that's a good question. So, definitely there are similarities in that TDP-43 is accumulating in the cytoplasm and creating dysfunction in a number of different aspects of the cell. And this pathological accumulation is also seen with tau in the case of Alzheimer's disease, and other FTD as well. A beta accumulates and forms extracellular plaques in Alzheimer's disease. So I think this toxic gain of function of these aberrant proteins accumulating is definitely similar. What I find really useful when I think about the Alzheimer's disease clinical trials is that they really paved the way to allow the ALS field to learn from what they did. So they had, many years of trying different anti-beta therapeutics, and they learned a number of important things along the way. One was to have the right biomarker that tells you that you're going into the right patient to treat them with that anti-beta therapeutic. Otherwise, the trial fails. The second is that you have to have different ways of treating, of trying to reduce A beta. So it could be an antibody approach. It could be interfering with the enzymes that ultimately produce A beta. And in the case of tau, it could be antibody therapeutics that intercept specific forms of pathological tau, or it could be an antisense oligonucleotide approach that's agnostic to the different forms of different kinds of tau fibrils, and rather can reduce theoretically, all those different pathological forms. And so I think the fact that the AD field took a number of different approaches and they were persistent to actually get to FDA approval of some anti-A beta agents is really a success in my mind. And the tau ASO data that's starting to come out from Biogen's clinical trials, I think, is also very optimistic. So again, I would say the really wonderful thing is that we get to learn from all of the things that they learned, but we're going to get to do this, I think, in a more accelerated pace.
Thanks, Amy. It's always great when you can learn from others and the experience that these other neurodegenerative diseases have had. So I'm seeing a lot of questions coming in, which is really exciting. I do want to note that if we do not get to your question, we will send an email and follow up post-event. But I'm just going to keep rolling along here. So our next question is, why does ALS diagnosis take so long? And is there any exciting research happening, like work on biomarkers to improve that diagnosis timeline? Amy, I'll start with you.
Yeah, I'll start. So this is the reason. It goes back to what I mentioned earlier. We just don't have a blood test right now. So if a patient walks into a physician's office and has, some fasciculations in their arm or some minor changes in muscle function, the physician can't just do a blood test and tell them that they have ALS. They have to go through a process of various clinical assessments to give the diagnosis by exclusion. ALS is a devastating disease, so it's kind of really the last thing you want to tell a patient. So physicians go through the process of understanding whether this could be an ALS mimic or some other disease that looks similar to ALS. So that can take a year or longer to go through that diagnosis. And what we've been doing is to really put a lot of funds behind biomarker development. There's some interesting things that have been emerging that we think have the potential to become an early diagnostic. And so if you look at our social posts, you'll find out more about some of the research coming from our funded grantees. But in addition to funding our grantees, that's part of the reason behind this large natural history study that we initiated back in 2021 is to collect blood and cerebral spinal fluid so that we can start to measure different proteins in those samples that might allow us to identify a biomarker that can be used early as an early diagnostic marker.
Thanks, Amy. And Manish, you have anything to add to that?
Well, I think Amy captured it perfectly.
Great. And so I think next we'll get into like this, what feels like the million-dollar question to me is, how do people get ALS? Is it genetic? Is it environmental? What are we looking at? Because we know, or at least right now, 10% is considered familial or inherited, and the 90% is sporadic. So, Amy, would you like to start?
Yeah, I can start with this. I think again, about 10% is inherited, but we've really only identified a few of the causal genes. There's probably room, not probably, there is a lot of room to identify additional genetic risk factors within that familial cohort and also within the apparent sporadic ALS, where mutations may occur de novo. So that's why we are putting such a big emphasis on generating whole genome sequencing data. Um, both short read and long read forms that allow you to get as much information as you possibly can from whole genome data from every patient enrolled in our natural history style and really study and enrolled in AGRI. So AGRI, our clinical study, has two parts that are complementary to each other. One is an in-clinic study where patients come in every 4 months, and part of that study is to collect whole genome sequencing. And also to ask the patients to fill out an environmental questionnaire. And the complementary piece of that is our in-community pop-ups, where we're allowing people who just are going to have a hard time accessing their ALS clinic, or maybe they have a language barrier or other barriers that prevent them from coming into clinic. We're going to them and we're collecting a one-time blood samples to collect, again, whole genome sequencing data in combination with their occupational and environmental history through a questionnaire. So we're going to be able to lock down, hopefully, in the future as we generate more data over the next 5 years from a goal of 6,000 ALS patients, what are some new genetic and environmental factors? And to date, I would just say there are some potential environmental risk factors that have been described. And so I would recommend going to CDC and publications from Palmetto, publications from the lead epidemiologists in the field. But some of those risk factors can include things like exposure to lead, smoking, exposure to heavy metals or agricultural toxins. And so again, I think the issue is it's just hard to quantify those exposures, and it can be variable across geographical locations. So this is why we're really invested in doing a large global scale initiative to find out more about these potential environmental and genetic exposures.
Thanks, Amy. And there's seems to be a question here that can piggyback off of this, and I'll pose this to Manish. So what are we doing to address the lack of access to research and support in the global south?
Yeah, thanks for that question, Stephanie. I think we have actually thought about this for a while, and we have obviously heard from the community about lack of opportunities. And I think one could put this in two broad buckets. One is engagement of the scientific and clinical community in the research that we support. And the second is the communities there who face ALS and creating opportunities for them to participate in ALS research so we can get to the solutions that we need for everyone. And sort of a longer term goal of creating conditions that they can also access clinical trials locally. So if you look at the first bucket, um, we support research in three ways. It's a research that we fund, which is obviously direct funding. And there we have reached out to the scientists and clinicians in different countries. Not all of them, and we certainly can use help of our global community friends to spread the message about Target ALS and the resources we bring to bear to support research. But where we have made contacts, we have encouraged them to apply for funding. We try to connect them to collaborators around the world so they can apply as part of collaborative projects. And then through our what Amy described global natural history study, we have got sites in now South America. We are looking at sites in onboarding sites in South Africa, Israel, Asia, Southeast Asia. So again, we obviously have a certain bandwidth and we can only actively engage so many sites in the natural history study. But the goal is to try to build those relationships and then from there branch out and try to engage communities in other countries as well and enable them to participate. In parallel, what we are trying to do is to build relationships between these clinical centers around the world, which we are referring to as Global South, and the pharma biotech companies that run clinical trials. And it's really important to first build those relationships and prove to these companies that these sites are capable of running clinical trials. And if they are not, that they have the opportunity to learn, identify gaps, learn, and fill those gaps. So they are prepared as these clinical trials come around. So that's sort of our intermediate to longer term goal to enabling these communities to benefit from all this work that is done and the output of it. And I know the Amy also mentioned the community-based pop-up clinics where we are going to the communities to collect samples. And again, the goal, at least we have, is that in all these communities that have so far not been able to participate, we actively reach out to them and lower barriers for them to be able to participate. Because as they become part of the broader community, they'll be able to get benefits from the output of all this work that is done as well.
Thanks, Manish. Yeah, it seems like there are multiple reasons why it's so important to like increase accessibility, both from gathering those patient samples, being able to run all of these analyses to figure out what those genetic factors are, what those environmental factors are, but also then again to encourage researchers around the globe to bring their ideas to Target ALS, and to support research and our vision of everyone lives, right? It's not everyone in the United States, it's everyone, no matter where they are. And so, really exciting that we're expanding Agri in that way. Another theme that's coming to the top here is about research on C9orf72. So, Amy, could you maybe explain that to the group, what it is, and also what we're doing to tackle it?
Sure. Thanks, Stephanie. This is a really important area of focus for us. I think people know that C9 runs in Dan's family, and so this is something that we've been trying to tackle in a number of ways. But just to start, let me talk about the lingo here a little bit. So C9orf72 is a gene that is encoded by nucleotides. Nucleotides make up all the genes in our body, right? And so normally, there are four different nucleotides that can come together in different arrangements and combinations to encode RNA. And RNA is that provides the template for proteins to carry out normal basic cellular functions that we need, right? So in the C9orf72 gene, what happens is that there's a so-called hexanucleotide repeat. So some of these nucleotides G4C2, they're occurring in repeat and they're existing where they shouldn't exist. So there's an abnormal repeat expression that occurs in this gene and it's expanded, meaning that if you have under 30 repeats, the likelihood of you having a healthy cell and a healthy life is probably pretty high. If you have expansion of those repeats, anywhere from, let's say, 50 repeats to even 1,000 repeats, this is correlating with ALS and FTD as well. And so the issue is here that there's complex biology. So we've learned this because we've learned from clinical trials from Biogen and some other pharma companies that targeting those aberrant RNA transcripts that are coming from that DNA hexanucleotide repeat and shouldn't be there and mucking up the cell activity. If you target the sense strand that forms, there's an antisense and a sense strand. That if you reduce that with an ASO, this just wasn't beneficial in patients. So this was a clinical trial that stopped about a year or two ago because there was no benefit in patients. And in fact, it was actually harmful to some extent to those patients. So the field has really been thinking about that outcome and trying to understand what's the right way to treat patients with C9orf repeat expansions. And so there are many different approaches that we're taking. And when I say we, that we're funding a number of different groups to figure out how to therapeutically modulate this. One way is to target the antisense strand. So we're funding a consortium between Ionis, Biogen, and Johns Hopkins to really study how we can target the antisense strand and whether that might be more beneficial than the sense strand. And so far, they're seeing some promising progress, and I'm sure we'll hear more about that from them in the future. The second way that we're doing this is we're funding investigators who are studying ways of directly targeting both the sense and antisense at the same time. So you're not just targeting one piece of the pathology, you're targeting both. And so there's a specialized way of doing that. And Kathy Morelli and some of the other scientists across our ecosystem are studying ways to do that and making really nice progress. Finally, there are CRISPR-based approaches that one could take to excise that repeat expansion from DNA. So we're funding Claire Cleland and some other folks on her team, and now in collaboration with Denali to really accelerate development of CRISPR therapeutics for C9. And I just want to mention quickly that I think these are all really exciting projects. I'm excited to see how they go. I think it might take another year or two until we see first in humans emerging from this work, but I'm very optimistic about this. And the other piece that we're funding is studying the basic biology and understanding or watching what's coming out of ALS research as a whole on C9 biology. So some pieces of information that are emerging are things like somatic expansion of C9. So meaning that it could be, and this is what researchers are investigating, that there could be differences in the length of that expansion. So it might be 1,000 or it might be 100 across different regions of the brain and spinal cord and across different cell types. And it might even be different between the brain and what we measure in the blood. So it's understanding what is the impact of that and how do we therapeutically modulate the instability of that expansion. And the other piece of information that I think has emerged that's important is that there can be fragility in the DNA around that repeat expansion. And so researchers are trying to understand what does that mean if there's fragility in the DNA region and it's affecting genes that are surrounding the C9orf72 gene. So it's complicated biology, but we've got a lot of different efforts focused on this. I'm sure we're going to see more from this in the near future.
Thanks, Amy. Clearly, there's a lot of work going on in a lot of different avenues to tackle ALS. And so we only have time for a couple more questions here. So looking forward, what research gaps or barriers are we planning to address next? What effort is most vital to moving the field forward? And I'll pass this one over to Manish.
Yeah, I mean, obviously, we're working on many fronts, and Amy has referred a lot of them. I think the one that I feel particularly excited about is related to the global natural history study and the community-based pop-up clinics and the other biospecimen efforts we have around postmortem tissue that we collect brain and spinal cord from people who have passed away after ALS. And what we are able to do now is generate large data sets from these samples in addition to distributing them to scientists worldwide. And things like genomic data sets, protein maps, maps of RNA molecules, and all of these are being made accessible to the research community worldwide through Target ALS data engine that Amy referenced. And so this, for the first time, is allowing data from different types of biospecimens to be integrated in one place and for scientists to visualize, mine, and analyze them to find drug targets and biomarkers for the future. And the way we look at this is that this is really the next inflection point for the ALS research landscape because we all talk about artificial intelligence, machine learning, but these types of technologies are only as good as the data that they are used on. And so we need a data set that is from as diverse a community worldwide and as rich and comprehensive as is possible. And that is what is being created for the first time through this effort that Amy referenced, AGRI, which is ALS Global Research Initiative that we launched. And I feel that as we build this up over the course of 1 to 2 years, that'll really, it has already created a momentum, but then we will have created a sort of galvanizing environment for this next generation of drugs and then drug discovery programs and subsequent clinical trials that'll truly have an even greater chance of success than we have ever had before. And that, I think, is just really, really exciting for us that we're able to do that. And these things require a sustained commitment. And we collectively have, as a foundation, are fortunate to be in a position and have made a sustained commitment to see through these efforts so that we can get successes on the other side.
Thanks, Manish. And I know we're coming close to time, but I would love to close on this final question. If there's one thing the ALS community can take away from the current research landscape, what would it be? And Amy, I'll start with you, but I'd like to hear from both of you on this.
Thanks, Stephanie. That's a great question. So I guess what I would just share with the community is that we should be incredibly hopeful that there are more breakthroughs coming. And the reason that we can be is just recognizing and reflecting on all the breakthroughs that have happened over the past decade or so. So just starting with the identification that TDP-43 is a common pathological hallmark in 2006, to understanding so identifying the C9orf72 repeat expansion is the most common cause of familial forms of ALS, to things like understanding utilizing technology. So the advent of antisense oligonucleotides as therapeutic modalities was demonstrated with nusinersen for SMA, and then translated over to show benefit in FUS patients treated with FUS ASOs, and then ultimately with Qalsody for the SOD1 patients. And finally, in there, you have this advent of neurofilament light and knowing that you can use this as a disease biomarker. It took quite a bit of time to generate data to convince people how we can use it, but it is being used well in clinical trials today and is quite informative. And so I think seeing just this really accelerated rate of new breakthroughs is really telling us that there's more to come. And we can already tell that these breakthroughs are bringing engagement from academics and biotech to ALS. At our recent meeting, we had 145 biotechs represented. So we're engaged. We have new technology to help us make these breakthroughs, and we've got a lot of activity on it. So, yeah.
Thanks, Amy. And Manish?
I would also use the word hope, but just a brief context on that is that I was talking to someone, a friend of mine, who was diagnosed with ALS about a year and a half ago, and he made this comment that maybe hope is a dangerous thing for someone with ALS because he said every time he goes to his clinician, they tell him about something more he has lost. And that did shake up my concept or framework of hope. But then I spoke to another person and they gave me a different framework, which I have held on to and which I'll share with you. So, what I learned was that hope is not the destination, it's the journey. And it is not a journey about necessarily having successes, but it is about a journey of doing the right thing for the right reasons because it's worth it. And that, I think, is the kind of hope that we, at least at Target ALS, like to uh, think of when we move forward with our mission. And that is what informs our mission with a sense of urgency always, but with deliberation so that we are making the right decisions always. And I'll also say that this hope is not just blind faith, because this, Amy mentioned earlier, this approval of this drug tofersen, which works for people who have the SOD1 mutation. Now, it is indeed only applicable to 1 to 2% of the genetic forms of the disease. But what it has shown us for the first time is that if we have the right drug target, people with this disease are showing reversal of symptoms. And that is a landmark for the space of ALS. So that gives us the hope that if we can find the right drug targets, this disease is a treatable disease. We just need to continue on the path that we are with urgency, with deliberation, and in a sustained manner.
Thanks, Manish. And thank you both for your time today. Really grateful to have you both on this call and to share your insights about Target ALS and the ALS landscape with the community. And thank you to everyone who joined us today and who's still sticking around, even though we went over. Thank you all for your questions, for your time. And as I said earlier, if we did not get to your questions, we will follow up with an email. We want to make sure we're being the best resources we can for you all. Thanks again, and I hope everyone has a wonderful rest of their day.
Thank you, everyone.
Bye.